that lecture I gave you in lab is the lecture for both lecture and lab. And as I said, you're responsible for that material both on the lab test tomorrow and also on the first lecture test, which will be the week after next. So, on that test we'll cover chapter 17 on the blood, chapter 20 on the lymphatic system, and chapter 21 on the immune system, which we have yet to get into. And as I said, the format of the exam will be short answer, fill in the blanks. Maybe one or two words, sometimes a brief phrase, sometimes a calculation. Now calculations, you're going to need to be able to show your work. Not enough just writing on the answer. If you need more space, put it on the back of the sheet. Okay? That's tomorrow. So, on the blood, by now you've had an idea, or you've had an opportunity to look at the blood cells, so you should be able to differentiate them, which you can expect to do tomorrow. So right now there will be smears on the blood, and you'll be able to identify the cells, be able to tell me what's their relative abundance, and also what do they do, what's their function. Okay? So by now you should be able to recognize these cells. It will not be from a projection, it will be from the microscope, or perhaps models. We've already been over the function of the blood, which you should know. We've been through the plasma, components of plasma, which remember makes up a little over half of the blood, and the different formed elements, which of course are the erythrocytes, the red blood cells, the leukocytes, which are white blood cells, and the platelets, or thrombocytes, which are cell fragments. We've been over disorders associated with red blood cells, also disorders associated with white blood cells, which you should be familiar with. The last time we were talking about blood clotting, which remember is part of hemostasis. Hemostasis is stoppage of blood flow. What's the first mechanism involved in hemostasis? Not blood clotting, but what? What? A vascular spasm. What is a vascular spasm? Okay, it's constriction of the blood vessel. And that's due to smooth muscle contraction in the blood vessel wall. We call that vasoconstriction. That narrows the lumen through which the blood flows. That reduces blood flow. It doesn't stop the blood, but it reduces the blood flow and thus restricts blood loss. We also talked about the second phase. What's the second phase of hemostasis? Platelet blood formation. When those platelets get activated, they stick together. They also stick to broken ends of the blood vessel, and they form like a temporary plug, if you will, called a platelet plug. And then the third and final phase is coagulation, or blood clotting. Remember, that involves a series of chemical reactions involving chemicals known as procoagulants, because they promote coagulation or blood clotting, also known as clotting factors. As I said, I'm not going to expect you to know all these, don't need to worry about the sequence, but remember they do react in a certain sequence. But what I do want you to know are the three phases of the blood clotting process. What's the first one? It's formation of an enzyme called prothrombin activator, or prothrombinase. That involves a series of reactions. And that can occur one of two ways: the extrinsic pathway or by the intrinsic pathway. You should know the difference. I don't mean that you have to list all the clotting factors in order, which is in detail in your textbook, but you should know, first of all, what starts it. What's the chemical that starts it? Number two, it involves calcium ions in both cases, and the end result, which is prothrombinase, a prothrombin activator. So what's the difference between the extrinsic pathway and the intrinsic pathway? There's two different ways to form the prothrombin activator. What starts the ball rolling? What initiates the extrinsic pathway? Tissue factor. If you want to be faster, you can say tissue thromboplastin. Right? What is it? Where does it come from? It's chemicals released from the damaged tissue outside the blood vessel. And we call it, when we talk about the extrinsic pathway, remember, this is critical. It means extrinsic to the blood vessels, because often when you got blood vessel damage, you got tissue damage outside the blood vessel as well. You get a cut, of course, you're cutting through the skin. You're cutting through the epidermis, which has no blood vessels at all. And then you go into the dermis. So if you got bleeding from the skin, you know you've at least cut into the dermis. No bleeding, just a scratch, literally, you're just in the epidermis. So extrinsic pathway, you got damage to tissues outside the blood vessel. So it's extrinsic to the blood, not extrinsic to the body. Make that distinction. The blood clotting that occurs outside the body is not by the extrinsic pathway, it's by the intrinsic pathway. Because intrinsic means intrinsic to the blood. Everything you need for blood clot is already in the blood itself. Okay? So tissue factor starts it going, involves a series of chemical reactions, involving these clotting factors reacting in a certain fashion, involving also calcium ions, and then prothrombin activator or prothrombinase is the enzyme. Okay, that's extrinsic pathway. Is it faster or slower than the intrinsic pathway? Faster. Why? Fewer reactions. Fewer reactions occurring. Okay? The intrinsic pathway, again, the end result is the same. Prothrombinase or prothrombin activator is formed. Again, it involves calcium ions, a bunch of clotting factors, even more now, reacting in a certain sequence. What starts it? Platelet factor, or PF3. This is something released by activated platelets, which are already in the blood. That's why I said, everything you need for blood clotting is actually already in the blood. So blood clotting occurs in a blood vessel where there's no damage to anything but the blood vessel is by the intrinsic pathway. And clotting occurs outside the body. You put the blood in, let's say, a tube, especially glass. It will readily clot unless it has been treated with an anticoagulant. A negatively charged surface stimulates the clotting process.
So are you saying the platelet factor starts at intrinsic path? That's what starts at intrinsic?
Intrinsic, yes.
Yeah, the intrinsic.
That's the instigator, if you will.
Okay.
It's released by activated platelets, not by tissues. But it works like tissue factor. Okay? And again, the end result is the same. Okay, that's the first step in blood clotting. But then we got two other parts of it called the common pathway because they occur in both. What's the next reaction then? Step 2, if you will. Conversion of prothrombin to thrombin. And it's catalyzed by what enzyme? Prothrombinase, or prothrombin activator. It's activating the prothrombin, converting it to thrombin, which itself is an enzyme. Okay? What's the third and final phase then? Conversion of fibrinogen, which is an inactive plasma protein, to fibrin. And that's catalyzed by what enzyme? Thrombin. Okay? Thrombin. And those last two reactions, converting prothrombin to thrombin and fibrinogen to fibrin, is called the common pathway, because that always occurs. What's different is how you form the prothrombinase. By the extrinsic pathway or the intrinsic pathway. When you get a blood clot forming an unbroken blood vessel, which sometimes occurs, that astounds most people. Why do we get a blood clot? Because you've got damage to the blood vessel wall. It may be not enough to cause bleeding, but you've got some damage. And certain diseases will do that. Atherosclerosis, for example, will cause damage to the lining of the blood vessel wall, and the result can be platelet activation and blood clot formation. So what pathways involved there? Got a blood clot forming an unbroken blood vessel. What's the pathway? Intrinsic. And again, intrinsic means not intrinsic to the body, intrinsic to the blood, okay, or the blood vessels, all right? And sometimes what happens is you invade the lining, you damage the lining. There's a condition known as atherosclerosis. You've probably heard about it. And a lot of people suffer from it, particularly if you got the high fatty diets of typical citizens of the United States and many Western countries, which is why a lot of us are carrying a little extra adipose, because we take in too much fat in our bodies. And what do? We store most of our calories as fat, okay? This can give rise to a condition known as atherosclerosis. You get these fatty plaques in the arterial walls. That causes damage. It also restricts blood flow. And with that damaged blood vessel wall, there's always a danger of blood clot formation, which can basically shut off blood flow. Occurs in the heart, you got a heart attack. Occurs in the brain, you got a stroke. Okay? You ever wonder, why do we store extra calories as fat? Why couldn't it be good old protein or carbohydrate? Why fat? Would it be nice if your extra calories were all protein?
Absolutely.
Not really. Why?
More calorically dense. You can store more energy in a small space.
That's right. Believe it or not, the fat per gram has more calories than either protein or carbohydrate. So if we stored most of our extra calories as protein or carbohydrate, we'd be even bigger. Okay?
Wow.
It might not be all muscle, it might be something else. But the point is, animals, since most animals or many animals are mobile, it's a lot easier to move a more compact body than a big one. So that's by the reason we store our extra calories primarily as fat. We have carbohydrate reserves, but mostly fat. Okay? More calories per gram. Now plants store a lot of their extra calories as carbohydrate, but it doesn't matter. Except in fairy tales, plants aren't walking around. Right? They're rooted, they're sessile, all right? But where is it critical for a plant to be compact? What part of the plant?
Seed.
Is where we get most of the fats from plants. The seeds. You got a seed the size of a boulder, it's a little hard to disperse the species, okay? So most plants have fairly small compact seeds, even the giant trees and so on, redwoods and sequoias and so on. Why? Because you can spread the seed much more quickly and much easier. Some of the seeds blow in the wind. Some of the seeds get picked up in your clothes or fur of animals and transmitted that way. And some, even better, you got something that's very edible around the seed, okay? The animal will eat it, and it often pass through the gut, undigested, and then stuck in the ground with a little bit of fertilizer. And often you'll see along a row, a power line, there'll be a nice little row of plants. Because the birds have been sitting up there, and of course, what are they doing? They're planting the seeds. Right? So, where do we get our oils and our fat and our fats from plants? Mostly from the seeds. A lot of calories, a lot of grams per gram, more compact. Okay, as I said, once that blood clot forms, though, get back on track here, that's not the end of the story, because first of all, the clot is rather a loose arrangement. You've got these fibrin threads, which is the end product of the blood clot formation. Conversion of fibrinogen, which is a water-soluble protein, to fibrin, which is water insoluble. But then what happens is some chemicals are released. This is called fibrin stimulating factor. And they're activated by the thrombin and calcium ions, which are already present. What that does is cause the platelets then to bind the fibrin threads more tightly together. That helps to strengthen the clot. But just to make sure, the platelets in themselves actually involve in what's called clot retraction or syneresis. They literally pull on the threads, and it squeezes the serum. The serum is the plasma without the clotting factor. So it squeezes the fluid right out of that clot, and now you got a big, thick mass without all the fluid. So it's not just a loose accumulation jelly-like. It's much more compact and much better at stopping blood flow. And then, not only that, but then the platelets also involved in repair. Once you've got the blood clot, remember that's temporary. You don't want it there permanently because that's going to restrict blood flow. You want to repair that vessel and get things back to normal. And the platelets are involved in that repair by releasing something called platelet-derived growth factor, PDGF. That stimulates growth in that it causes smooth muscle to repair itself. Remember, smooth muscle has the ability to regenerate, unlike most muscle in the body. It'd be nice if our heart muscle would do that, but it doesn't, unfortunately. So when you have a heart attack, that damage is permanent. That muscle is not replaced by more cardiac muscle, it's replaced by scar tissue, which is non-contractile. It'd also be nice if our skeletal muscle did that too. You say, well, my muscles get bigger when I exercise. That's true. It's not because you're getting more cells, it's because the cells are getting bigger. Okay? But a smooth muscle has an ability to reproduce. And it does here. And it's stimulated by this. In addition, it also stimulates the fibroblasts to produce more connective tissue. Remember the blood vessel wall, well, you don't know yet, but blood vessel wall, we're going to get in that soon, okay? Actually on Thursday, the exam will start the new unit. Blood vessel wall has not only smooth muscle in it, but it's also got connective tissue. Okay? And then the endothelial cells, remember these are cells that line the blood vessels, they reproduce and restore the line, which is very important, and a nice, smooth, swiftly line. Vitamin K, we mentioned that before, that's the coagulation vitamin. It's very important because the liver uses it to make many of the clotting factors. So if you're deficient in vitamin K, you may not make the clotting factors, and you may have trouble clotting your blood. This is why sometimes treatment with broad-spectrum antibiotics will knock out your normal so-called flora or normal microbiota. And some of these bacteria that we have in our lower intestine are very important because they synthesize some important vitamins, B vitamins and vitamin K. If you knock out these bacteria, then what happens is you may be deficient in vitamin K, and the result can be problems forming clotting factors and problems clotting the blood. And children especially are very susceptible to this. And vitamin K, like I said, is made by bacteria in the large intestine. It's absorbed and into the blood with our fats. And also, too, if you have fat absorption disorders, this also can result in bleeding because you're not getting the vitamin K, the liver's not making the clotting factors it should, and you have problems clotting the blood. Okay, once a clot is formed, you've stopped the bleeding, now the body wants to get rid of it. So there's an enzyme that does this. And the process, as a fancy name, is called fibrinolysis. Remember, it's fibrin that makes up the framework. Lysis means to break up or break apart. So literally, you're going to break up that clot. Fibrinolysis, then, is what's going to break up the clot at the site of the blood vessel injury once that blood vessel has been repaired. And there is actually, in the clot itself, what you need. It's an inactive form of an enzyme that's going to dissolve the clot. And this is called plasminogen or profibrinolysin. The active enzyme then is called plasmin or fibrinolysin. And this is actually, the inactive form is actually incorporated in the clot. And then once that clot then, it stimulates the endothelial cells, which line the blood vessels. They release what's called tissue plasminogen activator, TPA for short. And this is what converts this inactive form of the enzyme, plasminogen or profibrinolysin, into the plasmin or fibrinolysin. And this is what literally catalyzes a reaction to break down the clot. So you've got this inactive enzyme already present in the clot. Once it's stimulated, and it's stimulated by the cells lining the blood vessels, the endothelial cells, then it becomes active and it dissolves the clot. It catalyzes a reaction to dissolve the clot. So it's very nice. You got built in the mechanism right there, or the chemical right there, is just an inactive state. And once it's activated again by these endothelial cells after the vessel is repaired, then the clot is usually broken up, dissolved. Okay. And also thrombin itself and another clotting factor work together. When they're activated, they can also convert plasminogen to plasma. And again, plasminogen is the active form. It's also called profibrinolysin. The active form then is called plasmin and fibrinolysin. Okay? Because that's what it does. It lyses or breaks up the fibrin again are the threads that hold the clot together. And the plasmin then will inactivate clotting factors, including the prothrombin and fibrinogen. So you're basically not only breaking up the clot, but you're stopping formation of more clots. You want to get rid of the clot. Any questions on this? As I said, the toughest part of this unit, I believe, in Chapter 7, is knowing the clotting factors, knowing the anticoagulants, and what they do. So I would strongly suggest, when you're out there having fun in the sun, okay, you might want to take a little time and go over these things. Maybe with flashcards, or maybe a chart, whatever helps you learn. You're going to need to know these. Okay. Any questions on this so far? Let's look at control measures, hemostatic control measures. Once blood clotting is formed, you're going to have some way to control it. That's what we're talking about here. First of all, doctors said we limit blood clot formation. One is to remove the clotting factors. You can basically keep them apart. You're not going to have the clotting process occurring. So they can literally be washed away. And that's why where blood clots most likely will form are in areas where there's little blood flow, and the blood flow is relatively slow, like in the veins and so on. Where you don't get clotting would be in the major arteries. If you sever a major artery, you'll bleed to death before it ever clots, because the clotting factors just can't accumulate and they can't react. They get swept away. That's why with arterial bleed, very serious. You basically have to derive direct pressure, or you go upstream to a pressure point and clamp off the artery. And temporarily you can do it with a tourniquet. And if you do that, of course, you don't keep it permanently on, otherwise the tissue is going to die eventually. So you've got to release that tourniquet occasionally. And of course, the proper thing is get them to medical attention as quickly as possible. 911. Okay? Okay, also inactivation or inhibition of these activated clotting factors, there is a compound known as antithrombin 3 or AT3 for short. This is actually a plasma protein. We will inactivate the thrombin. Remember, thrombin is an enzyme. It's one of those clotting factors. You want to get it out of the picture. Another one is called protein C. This is produced by the liver. That, along with antithrombin 3, or antithrombin 3, will inactivate other clotting factors. So one thing is you can sweep the clotting factors away with fast or rapid blood flow. The second is to inactivate clotting factors. Both these can help to control and limit the formation of blood clots. Also, there are factors that basically inhibit undesirable blood clotting. The smooth lining of a normal blood vessel, which is formed by those endothelial cells, it's called the endothelium collectively. This prevents the platelets from becoming activated. They don't aggregate, they don't form the platelet plug, they don't release the chemicals to stimulate the blood clotting process in the first place. And that's why that lining is so important. That's why, too, when the lining gets ablated with these fatty plaques forming, okay, atherosclerosis, there's always a risk of blood clot formation. And again, that blood vessel may not be leaking, you only have damage to the lining and part of the wall, not enough to cause the blood to flow out, but enough to activate platelets and bring about blood clotting. Another thing is production by the endothelial cells. I mentioned this before, of prostacyclin, PGI2. That's what limits platelet clot formation. If you can limit that, limit the activation of platelets, you can limit blood clot formation as well. And third, endothelial cells actually can and do secrete heparin, which is an anticoagulant. So we got cells in the body that actually secrete heparin. Cells lining the blood vessels, endothelial cells, and also cells called mast cells, which we will talk about later. Mast cells are actually involved in the immune system. Very similar to basophils, but they're not in the blood, they're outside the blood. They too, like basophils, produce heparin. Basophils produce heparin, mast cells produce heparin, endothelial cells produce heparin, an anticoagulant. Any questions? Okay, let's look at disorders associated with blood clotting. So hemostatic disorders. One thing is blood clotting that occurs in unbroken blood vessels. We call this intravascular blood clotting. And the clot that's so formed is called a thrombus. The plural form is thrombi. That's where we get the term thrombocyte, another name for platelets. So again, a thrombus is a blood clot that forms in an unbroken blood vessel. But the fact that it's unbroken does not mean it's not undamaged. It is damaged. And that's what brings about the blood clot formation. And again, what this can do, of course, is restrict blood flow. And to a vital organ, it can be critical. If that clot breaks free and circulates with the blood, it can be even more dangerous. That's called an embolus. The plural would be emboli. And sometimes this occurs. That's always a risk with surgery, is blood clot formation. And especially when you have blood clots that get into the vascular system and they start being carried by the vascular system, and they usually end up in some smaller vessel and sometimes in a vital organ, which can shut off blood flow. So pulmonary embolism or whatever, this can be very serious. And that's always a risk with surgery. There are two big risks with surgery. One, of course, is infection. Sometimes a person goes in, the operation is a success, but then they come out with a nasty infection. And the second is blood clot formation, which sometimes shut down blood flow to vital organs. But other things can also act as emboli, not just blood clots that are broken free from site of formation, but also debris. Sometimes gas bubbles, we talk about that, remember, with the Benz, Caisson's disease, nitrogen bubbles come out. Okay? And also debris like a piece of fat and so on that can be transported and become blood vessels. Okay? Any questions on this? But again, it's critical that the blood vessel be intact, and it's very critical that that lining remains unabraded. Because once it does, there's always a risk of platelets being activated, and once they're activated, a good risk of blood clot formation. Any questions? They actually break up the blood clots, and they can be injected basically into the body and reduce the severity. That's why, too, if a person has a heart attack or a stroke, you can get them to medical attention is very important. The sooner they can get medical attention, the better. And the less likely the damage, or the less severe the damage will be, especially with heart attacks. Because what they'll do is basically put these toxic substances in, and this can break up the clots and help to restore blood flow to agents. And one, for example, is streptokinase, I call streptase basically, comes from actually a enzyme that is produced by bacteria, Streptococcus bacteria. These bacteria, many of them are pathogens, and what they do is they use this to break up blood clots so they can spread out in the tissues. But one way, when we have an infection, we try to localize it, and one way we do that basically is to form blood clots around the agent and kind of wall it off. The bacteria can fight back by producing these agents that will break up the clots, like streptokinase. So we have purified forms of this basically we can use actually as clot-dissolving substances to treat people following a heart attack or stroke to lessen the severity and to help to restore the circulation by breaking down those clots. Any questions? Okay. Take a look at some bleeding disorders. First off, thrombocytopenia. Thrombocytopenia means you've got a deficiency in your platelets, or thrombocytes. The result is, of course, you can have bleeding resulting from that because you don't clot the blood as you should. And this is usually associated with a platelet count of less than about 50,000 per cubic millimeter of blood. Normal platelet count is usually around quarter to a half million. So they're fairly numerous. Okay? So if it's really low, the result, of course, is you don't clot the blood as you should, or as fast as you should. So you're going to have bleeding. That's called thrombocytopenia because of the reduced numbers of platelets. What can bring this about? Well, anything that damages, destroys, or suppresses the red bone marrow, because remember, platelets, like the blood cells, are also made in the red bone marrow. And this can be in various drugs that are used, cytotoxic drugs. Can also be used, or can be the result of radiation, which is sometimes done. So some therapies result in radiation or use of cytotoxic drugs, particularly cancer therapies. Or it can be bone marrow cancer itself and also causes too. We mentioned like where you have leukemia. Leukemia, of course, affects formation of blood cells and also formation of platelets. Something else that can bring this about is that the liver is not working as it should. Remember, the liver, the major source of our clotting factors. So impaired liver function. This can result in a deficiency of the clotting factors that are made by the liver. And what can bring this about? Well, hepatitis, sometimes due to chemicals. Because one of the things the liver does is detoxify chemicals. Another thing is hepatitis due to a viral infection by hepatitis virus. Hepatitis A, hepatitis B, hepatitis C, hepatitis D, hepatitis E, there are a bunch of them. Okay? The most dangerous, though, are B and C. There is a vaccine for A, that's usually mild hepatitis A infections, usually no severe or permanent liver damage. Hepatitis B could be a different story. There is a vaccine for that. If you're going into medicine, it's a good idea to have that, and you're going to contact patients who have hepatitis. To my knowledge, there is not yet a vaccine for hepatitis C, but that can also be very nasty as well. Both B and C can cause permanent liver damage, particularly when they're in combination with another hepatitis virus like hepatitis D or hepatitis E. And they frequently give rise to cirrhosis of the liver, where you have scar tissue forming, or they can predispose you to liver cancer as well, increase your risk of liver cancer. And also cirrhosis. And cirrhosis, again, is where we get the formation of scar tissue in the liver. Now, the liver has a great ability to repair itself, but you can overdo it. Now, one thing you can overdo it is chronic alcoholism. And this is why many alcoholics, okay, end up with cirrhosis of the liver, and sometimes it's fatal. It can be very serious. And, of course, also vitamin K deficiency. But vitamin K, like I say, the liver uses that to make maybe the clotting factors. And again, this can be tied to destruction of the normal bacteria that are found in the large intestine. very susceptible to this are infants and young children. Treated with broad-spectrum antibiotics can knock out their flora. These are useful bacteria, and then the result can be you don't get the synthesis of these vitamins that the bacteria make, and you're not absorbing enough. Because we can't make these vitamins. Bacteria do. Like I said, we have bacteria in our bodies that are very useful. Not all bacteria are bad. But when you lose these bacteria, they can be bad. First of all, you don't have what they do, the vitamin synthesis, and secondly, their place may be taken over by a pathogen that can cause severe disease. And a good example is C. diff. You may have heard of it. Clostridium difficile. It's quite common in many clinical facilities. It's very contagious, can be spread readily from patient to patient, and it causes a raging gastroenteritis. And it's not uncommon to see it following broad-spectrum antibiotic treatment where you knock out the normal flora. The C. diff is there, but in small numbers, contained by your normal bacteria, and then suddenly you have a wide-open environment, and it just multiplies. And it can be very difficult to get rid of.
If you diagnose with cirrhosis of the liver, it can't repair itself once you've been diagnosed with it?
With what?
With cirrhosis of the liver.
Well, yeah, if you give it time to repair itself, yes. If you're a drinker, obviously the thing is you want to cut back on the alcohol.
And
That's not
increase
very
the vitamin
common
K.
cause.
Can you increase the vitamin K? That'll help too?
Well, you can if you restore the flora, the normal flora, intestinal flora. That's a problem, though. Antibiotics, we use antibiotics, unfortunately, indiscriminately, too much. We often use them when we shouldn't be using them, and then we overdose on them. And what you're basically doing when you use an antibiotic is you're trying to knock down the bacterial infection. It doesn't work for viruses. So your immune system can take over and destroy it. If you don't follow the regimen and you don't get a therapeutic dose, it's not going to work. And all you're doing basically is selecting for resistant floras.
What do the probiotics do that you can use?
Probiotics is basically to help you restore your normal flora and
So usually,
facilitate the
that's
growth.
not something you should take as a vitamin or something? I hear
Well,
people are telling you all kinds of
there are
stuff.
all sorts of stuff you can buy. Okay, a lot of it is, I'm sorry, it's not going to help you that much.
That's what I thought.
A good normal diet is less expensive and can accomplish the same thing. So all these supplements, okay? I'm not saying they're not good, they can be, but overindulgence, good is, that can be very good, but more may not be better. It's sometimes harmful, okay? So beware. And some vitamins, okay, you overdose on the vitamins, what do you do? They end up going down the toilet. You get rid of them. You flush them out of your body, especially the water-soluble vitamins. Okay, like B and C and some of these. Some like A and D and so on, you get hypervitaminosis where actually can cause damage.
Oh,
Overdose.
really?
Yeah. So beware. More is not always better.
Okay, Doctor, scar tissue. Is that just the scar tissue? Is it damaged cells in the liver?
What?
Cirrhosis causes.
Yeah, cirrhosis basically you've got destruction in the liver and it's replaced by scar tissue. That's cirrhosis.
So scar tissue is the healing part of the.
Well, scar tissue is interparts connective tissue, like a scar.
Right.
Okay? And if you get too much, basically, you can overwhelm the liver, and the result, the liver shuts down, and then you're in trouble. You can't live without a liver. The liver has a good ability to repair itself, but like I say, you can overdo it. And one prime way is, of course, with overindulgence in alcohol, because alcohol is toxic.
Is there something besides alcohol that people get cirrhosis from?
Certain drugs can do it. Certain drugs are hepatotoxic. The classic one is acetaminophen.
Oh,
That
really?
was tried on TV, but
What is it?
acetaminophen, Tylenol, that's the active ingredient of Tylenol. They'll say, well, don't take aspirin, or don't take ibuprofen because it causes GI tract bleeding, which it does, okay? So take Tylenol. Well, Tylenol also has a downside too. It's hepatotoxic. In high doses over a prolonged period of time, you cause damage to the liver. Okay, so beware. And on the thing here with the liver, I remember seeing a program, it was probably one of these, Dr. G, you've probably seen it, and she's basically a forensic pathologist, and she gets these cases in, of course, her job is to determine the cause of death. I remember bringing this one in, and this guy, they got him from his apartment, and of course he was dead, there was blood all over the place, so obviously they suspected foul play. So they brought him in, and she examined the body organs, and she found his liver was completely shut down. He'd been a chronic alcoholic, and and he basically destroyed his liver to the point where it was no longer synthesizing the clotting factors, and he literally bled to death. Just hemorrhaged. The liver basically shut down. It wasn't making the clotting factors, so he couldn't clot the blood, and he basically just bled to death. And not just internally, but through various openings in the body as well. And that's why the blood was all over the apartment.
Wow.
So, yes. Yes.
My dad just had a liver transplant because he had cirrhosis. He's not alcoholic, but now that he has his new liver, he's still having like clotting issues and he's built in. Is that to take a while for your body to like
Yes, it will.
readjust?
Yeah. If somebody has that, you definitely don't want to be drinking.
No, he doesn't drink.
Yeah, that's good.
He never did it. They don't know why.
Well, some people, the liver just goes bad for some reason. Some are predisposed to certain things too. And some chemicals in the environment, sometimes chemicals in the environment too, that you pick up. And there are a lot of things that you can pick up. Well, as I say, one of the things the liver does is to detoxify things. And you can basically overwork it. And one of the big toxins that's very common, of course, is alcohol.
Where does the alcohol go? Like they say the liver can only process 1 ounce per hour or whatever. Where's the
It
rest
depends
of
on
it?
the person, and it depends on your body size. Obviously, a smaller person, you're going to get the effects.
Is it just waiting to go in there and there it just kind of goes wherever?
Yeah, well, moderation is the key. Yeah, you get it up here, that's the effect that we like, okay? But when you affect the other organs, that's the effects we don't like, and it can have an effect on various things, especially the liver. But again, it's highly variable. Some people can process it better than others. Part of its body size. A bigger person generally can handle more than a smaller person. Males more than females. That's why I say, females shouldn't drink more than this, males more than this. And usually it's more for the male. But it varies, the person. Okay. Another body disorder I'm sure you've all heard of is hemophilia, the bleeding disease. Now, hemophilia is more than one disease, but these are hereditary. This is something you inherit. You don't pick it up by contagion, okay, by being close to a hemophiliac, person who suffers from hemophilia. You get it basically from your parent, particularly from the mother. And what it is basically, the signs and symptoms in various forms of hemophilia are very similar, but they differ in severity. And it depends on what clotting factor is absent or not abundant enough. Hemophilia A, sometimes called classic hemophilia, this is the most common form, and it's due to absence of a clotting or deficiency in clotting factor number 8. Hemophilia B, it's a little less common, is due to deficiency in clotting factor 9. And hemophilia C, which is a much milder form, due to deficiency in clotting factor 11. So different clotting factors involved here. But in all cases, you've got a deficiency in a clotting factor, and this slows down the blood clotting process, which means that you will bleed. And often minor injuries can be very severe bleeding. Sometimes just you bump against something. Most people get a little bruise, may bleed a little bit, and it stops. Okay? But for the hemophiliac, they can bleed for hours and hours, or a minor cut can result in this. And these two forms, hemophilia A and hemophilia B, are said to be sex-linked. And we'll talk about this later in the course we do in genetics. But sex-linked means that the gene is carried on the sex chromosomes. And in humans, you probably know, a normal human female has 2 X chromosomes. A normal human male has an X and a Y. The trait, though, for hemophilia A and B, the gene is found only on the X chromosome. And you see right off, women carry 2 X chromosomes. So for a woman to be a hemophiliac, she's got to have the recessive form of the gene on both of her X chromosomes. By recessive, I mean it's only going to be expressed when it's with a number like recessive, because the trait is due to recessive. Okay? Males, whatever they have, be it a dominant or recessive, they're always going to express it. And that's why you see a lot more males that are hemophiliacs than females. Which is fortunate if you're a woman for a very good reason. What happens when you go through puberty? Your menstrual cycle starts. For most women, okay, that bleeding is, it's annoyance, but it stops. For a woman who's a hemophiliac, she could bleed to death. So what they often would do is basically do a hysterectomy before she reaches puberty.
Wow.
So she doesn't have that. Very severe. Unfortunately, again, we don't see many female hemophiliacs. It's mostly males. But the female will carry that recessive trait, perhaps on one of her X chromosomes. She doesn't express it, but who gets it?
The male is on.
The son. The son gets his X chromosome from his mother, and the Y chromosome for maleness from his father. So whatever's on that X, be it a gene for normal clotting or one for the disease, he will express it. This also applies to red-green color blindness, which is an X-linked trait as well. That's why you see a lot more colorblind males than you do females. Now that's not as severe. You can memorize the traffic pattern and lights and stuff like that. And to a male, okay, who a person with this red-green color blindness, you see shades. You don't see the color, but you see different shades of gray. Okay? But hemophilia is a different story. That can be very severe. And in some cases, it can even affect the outcome of history. So I'm going to just get off on a tangent a little bit here. This will not be on the exam, okay? Back around the time of the First World War, many of the families, or I should say many of the countries in Europe were ruled by royal families. And unfortunately, a lot of the royal families, the members, were related by blood. Queen Victoria, for example, was a reigning monarch. This is before the First World War, okay? She was a carrier for hemophilia, which nobody really realized. She, of course, wasn't hemophiliac, but she spread it. And some of her offspring, some of her granddaughters and so on, were also carriers. And one of them happened to be the wife of the Russian Tsar. Now, at the time, the First World War, the Russians, of course, along with the British and the French, were fighting the Germans and the Austrians. And many of these leaders of these countries were related, okay? The British king, George V, was related to the Russian Tsar, Nicholas II, okay? And of course, they were on the same side. But they were also related to the Kaiser, who was the Emperor of Germany. Let's make a little shorter story of this. The Russians, of course, were fighting the Germans on the Eastern Front, and they were getting stomped by the Germans, okay? In the meantime, of course, the Russian Tsar and his wife had 4 children, 3 daughters, and then the heir apparent to the throne, Nikki. But unfortunately, Nikki had hemophilia. And the doctors at the time didn't know much about it, and they could not control the bleeding episodes. But the Tsarina, the Tsar's wife, was introduced to this monk called Rasputin, who was a despicable character. Okay? He was a lecher, he was a drunk, a womanizer, just a terrible person. But he was a monk. And for some reason, he could control the bleeding episodes. There was something about him. So obviously, the Tsar's wife was attracted to him because he could help her son, which the doctors couldn't. So she wanted him around. But he was giving the royal family a black eye because, first off, the Russians were suffering terribly because of the war. They were getting clobbered by the Germans, and it was a poor country to begin with. And you had the haves, the Romanov family, okay, and then the peasants were literally starving to death. So it was ripe for revolution. What the Germans did, of course, is they wanted to get Russia out of the war. So they brought a man by the name of Lenin, smuggled him into Germany, and of course Lenin helped to bring about the Russian Revolution in 1917. And he promised that if the revolution is successful, I will take Russia out of the war. So the Germans were all for that. It would mean they wouldn't have to fight on one front, and they could concentrate on the British and French on the Western Front. But in the meantime, this Rasputin character, like I say, was giving the family a black eye. They were already in trouble with the common people because of disparity. Well, then to make matters worse, because things were going so bad on the Eastern Front with the Germans, the Tsar decided he would take control of the forces, which was a bad decision. Number one, he wasn't really a military commander. He should have left it up to his generals. And secondly, he left the reins of the government in the hands of his wife. And his wife happened to be a German princess, which didn't help matters either, because of course, who were the Russians fighting? The Germans. So they felt she was kind of maybe a traitor anyway. We don't want to trust her. And then having this character around Rasputin made things even worse. So the Russian family, or a lot of the princes that related, said, we got to get rid of this guy because he's just making things worse. She said, no, I want him here because he helps control the bleeding episodes. So they said, all right. So they invited Rasputin to an all-night drinking party. They got him drunk. They fed him poisoned wine to kill him. Didn't work. So they shot him. He was still alive, so they put a rope around his neck, hung him, and then dragged him out and threw him in the river. Well, later on, they found him. He was dead eventually, but his fingers were basically clawed to the bone. Obviously, he was still alive when they threw him in the river. He was trying to claw his way out from under the ice. But he had made a prediction. He said, if any member of the royal family has anything to do with my death, the dynasty will fall, which of course he did with the Russian Revolution. Not that that caused the Russian Revolution, but it contributed, if you will, in a very small part. Because of their son, who was a hemophiliac, and the mother, the doting mother, trying to help control the bleeding episodes by bringing this disreputable character to do it, and giving the family even worse press, basically, because of what she was doing. So, in 1917, in the fall, of course, the Russian Revolution took place. The Tsar and his family were basically put under house arrest. They were sent out to Siberia, eventually brought back because the country was in turmoil between the Bolsheviks, the Russian communists, and the Mensheviks, who were trying to restore basically the monarchy. So the Bolsheviks realized they had to get rid of the Tsar because he was a focal point for the Mensheviks, the non-Russian or the non-communist Russians. So they executed him and his family.
Wow.
And you may remember a person by the name of Anastasia. That was the youngest daughter. And a few years ago, I think about 10 or so years ago, this woman comes forth and she claims that she is Anastasia, the long-lost daughter of the Russian Tsar. Well, it turns out when they did DNA tests, she wasn't. But she, too, had been executed with the rest of the family. Anyway, if you're interested. Probably not, but this will not be in the test. But this is actually something that occurred. Like I say, sometimes a disease can play a role, even a minor role, in some of the events that occur in history.
How was he stopping her with the episodes?
What?
You didn't explain how he was stopping her with the episodes when she had to do it.
Nobody knows. Nobody knows. He was very charismatic. He had that effect on a lot of people, and that's why a lot of people thought he was just a charming character. You know, even though he was dirty, he was unschooled, he was a lecher, he was a drunk, a womanizer. But for some reason, he had just this magnetic spell over a lot of people. And he was self-taught, like I say, but he was semi-literate, maybe semi-literate. I don't know. Nobody really knows. But some people have that kind of a magnetic personality. If you look at pictures of his eyes, you know, it's sort of like a Charles Manson type thing. You know who Charles Manson is?
Oh, yeah.
Okay. Well, he's still in prison, fortunately, and he still thinks what he did was right. You know, he doesn't feel any remorse for the murders that he helped to bring about. Anyway, some people are like that, and I don't know. I don't know about this or not.
That seems really far-fetched to poison him, shoot him, hang him,
Yeah.
throw him underwater.
Yeah. But actually did happen. Okay, getting back here to what we were discussing. We'll get off this tangent. Okay, human blood groups. Now, we've already done this in the lab, so this should be basically a review. Blood types, of course, are what we're looking at here. There are many different human blood types, actually. What determines a blood type is the antigen on the surface of the red blood cell. That's called an agglutinogen. And these react with antibodies called agglutinins, which of course are in the plasma. Remember, antibodies are gamma globulins. They're part of those plasma proteins that are found in the plasma. And when the antigen and antibody react, you get these complexes. With the antigen and antibody cross-linking, you get these big clumps, basically red blood cells with the antigen on the surface held together by the antigen. And we get these antigen antibody complexes. And by the way, AG stands for antigen, AB stands for antibody. And as I said, and you've seen this in lab, this is how we type blood. We take a typing serum that has a known antibody in it, you react it with a blood type, or a blood rather, and if you get agglutination, that means that antigen is present in the blood. So to determine what antigens are present, and which are not, you can determine the blood type. It's exactly what you did in lab, or at least you simulated, or what have you.
We didn't see one that was O, where it says neither A nor B. How do you get type O?
Well, think of type O as 0, neither A nor B. Okay, so here we are. Type A, the antigen is present on the red blood cells. And by the way, there are over 100 different known erythrocyte antigens, but many of them are familial. They're only phosphorylated by blood, but there are about 30, 35 common antigens that we see in most human blood. The only two we're going to look at are the ones that are responsible for most of the transfusion reactions when the blood is mistransfused. And that's the ABO group and the RH group. Okay? So let's look at that. Type A blood, you got the A antigen on your red blood cells. you've got the anti-B antibody in your plasma, because B antigen is a foreign antigen. And you could donate, obviously, yourself, type A, and to type AB, small quantities. Can receive from itself A and small quantities of O. Type B, the antigen present on the red blood cell, anti-A antibody, which is again for a foreign antigen, and again can donate to itself in small quantities AB, receive from itself, and also small quantities of O. Type AB has both A and B antigen, neither antibody, neither anti-A nor anti-B, and it can only donate to itself, AB, but it can receive from any of the 4 blood types small quantities. Okay, but again, small quantities, usually a unit or less. And type O, think of it as 0, neither A nor B antigen present, but you've got both anti-A, anti-B antibodies, because A and B are foreign antigens versus the type O. And small quantities, you can donate to all the blood types, but it can receive only from type O. And the key is this: the recipient's plasma must not have antibodies to the antigens on the donated red blood cells. Otherwise, you agglutinate all those cells, eventually they're lysed, and then the hemoglobin spills out, gets trapped in the kidneys, and can cause severe kidney damage. And that's where usually the problem is with a transfusion reaction. Damage to kidneys and sometimes death as a result. And it shuts down the kidneys, or can shut down the kidneys. You say, well, how can O be a universal donor when it's got antibodies to both A and B? Because in small quantities it's okay, because the vast quantity of the blood of the recipient simply diffuses the type O blood, so it's not severe. But if you had a massive transfusion, it's got to be the same blood type, because then you'd have all this antibody from a massive transfusion of O, and you get a lot of antigen-antibody reactions. Okay, so only small quantities in OB. And a true universal donor would be what? O and O negative, okay? Not O positive. Negative means you don't have the antigen, the Rh antigens. Positive means you've got those antigens, which are foreign if you're O negative, foreign antigens to you.